All right, peace be upon you all, and welcome to another episode of Seeing God in Biology with Dr. Fuz Rana. And today he's going to speak to us about even-bit parity code. And, well, this is a very familiar term to me as a communications engineer because we use this for error detection, sort of. But I never knew that this is something that we can really find in biology. Fuz, how can this show us God's fingerprints in biology? Can you give us a little teaser about that?
Sure. Well, to me, one of the most mind-boggling things I've ever learned is that embedded in the structure of DNA is essentially an even bit parity code. And this was the
Wow.
insight of a scientist from Trinity University in Dublin, Ireland, and it's used in a sense as an error detection mechanism. And to me, it's just mind-boggling because it means that not only is there digital information built into the structure of DNA, but the way that that information is organized and the way that that information is operated on by the cell's machinery bears an eerie similarity to the way that we would organize and operate on information in computer systems. And that to me is highly suggestive that there's a mind behind everything.
Absolutely. But let's first explain to the audience what parity checking and parity bits are. So in my discipline, in communications and in information systems, we have typically two parties, so a sender and a receiver. So the sender sends a piece of information to the receiver, but there is a channel in between where information can change or errors occur. So the receiver cannot guess if the information that he received are essentially the information that the sender has sent. And parity bits is like those extra bits of information where we would both agree on a protocol so that the value of the bit can be checked versus the data, and then the receiver can know. So there is two types, even and odd parity, and they're essentially almost the same. The difference is in the protocol only. So I have a slide here. Actually, it's a simple thing. So let's assume that we're sending 7 bits by 7 bits, and the 8th bit, which will complete 1 byte, well, a byte is 8 bits, would be the parity bit. So if we agree on a protocol that we are following, for example, even bit parity, then the receiver will expect that the complete byte received has an even number of ones. Okay. So, for example, in this example, if the original data is 1001001, then there are 3 ones. Then the sender will insert the parity bit so that the total number of ones becomes even. In this case, these are 3 ones. So an extra 1 will be added to the extent that the whole byte will have 4 ones. Now, if one of the bits was changed due to noise or due to communication errors on the channel, so one of the zeros, for example, changed into a 1, then the receiver will now receive 5 ones. And he knows that the total number of ones due to the discretionary setting of the parity bit to complete even, now the total number of ones will be 5. Then he will know that the message must have been corrupted, and the receiver will request a resend from the sender. Also, for example, if one of the ones was changed to a 0, then you will have only 3 ones, and then the receiver will also know that something has went wrong and will request a resend. Obviously, this can, if 2 bits change, then maybe even bit parity or parity will not detect it. But there are other tricks, for example, to have a parity block instead of just a parity bit. But using parity, we can not only detect whether there is an error or not, but we can actually, using little bit more tricks, actually know which bit exactly has changed if we have a parity block. So there is further extensions to that. And this is on this slide, you can see that if we agree that the protocol is even bit parity, so in this example where there is 3 ones, the sender will add an extra 1 to make them even number of ones. If it's odd parity, then the sender will just keep it 0 because there is an already odd number of ones. And if the sender and the receiver agree on the same protocol, then the receiver will always know if the data that has been received if there is a fault, it will definitely know. if even parity checks okay, then it's likely the same, unless a sophisticated kind of double errors has occurred or something. So this is what engineering or information systems or communication engineering is using, even parity. And this is definitely, if somebody shows me two parties that are communicating and the receiving party is checking on the parity, then those guys must have agreed. There is a work of a mind there for sure. So now I'm pretty excited to see how this is being used in biology. So let's see.
Yeah, and I by no means am I an expert in information systems or communication theory, but my understanding is that a parity bit mode of error detection, as we've described it, is probably the simplest, most straightforward way to do it. And as you mentioned, there are clearly much more sophisticated, techniques that are available.
Yeah.
So this is really ideal for error detection as opposed to error detection and correction. But as I mentioned, this concept is actually manifested in the structure of DNA. And maybe what we could do is just very quickly review the structure of DNA. We put together a short video, about 20 minutes long, where we kind of get into some of the details of the structure of DNA that viewers may want to review if they're not familiar with the structure of DNA. But just a very quick review by means of giving us some some starting point for the conversation. And so this is a diagram showing three different depictions of the DNA molecule. And I like to say that DNA is kind of like has a ladder type of geometry, where the backbones of DNA are like the uprights of a ladder. The rungs are like the side groups that extend from the DNA backbone. And if you twist that, you get in a right-handed way, you get the well-known DNA double helix. Well, when we look at the structure of DNA, again, it's two strands of DNA, or the DNA is made up of two molecular strands that essentially align with one another. And the interaction of the side groups is highly specific. There are 4 different nucleotides that are used to build DNA: adenine, guanine, cytosine, and thymine. And when the side groups of the two chains interact, adenine is always interacting with thymine, and guanine is always interacting with cytosine. And that interaction is very precise. This is an incredibly sophisticated molecular recognition system. When we look at the alignment of the DNA molecules, and that is actually established through what are known as base pairing interactions. And so in the top of this slide, we're now just visualizing are the side groups of each of the nucleotides that make up DNA. These are known as the nucleobases, and these are found in the interior of the DNA double helix. And what we can see is that when these two bases are aligning with each other, you have a total of 3 rings that make up that span the space between the two DNA uprights, or the two DNA backbone strands. And so when the two side groups of the two different strands interact, they interact in such a way that you have a total of 3 bases, so that the distance between the two strands is more or less constant along the length of the double helix because of that. And so guanine always pairs with cytosine. Guanine has the side group, the nucleobase, is a fused 5-membered ring and a 6-membered ring, interacting with cytosine, which is a 6-membered ring. And then adenine, likewise, is a fused 5-membered and 6-membered ring that interact with thymine, which is a 6-membered ring. But
so this causes the whole thing to be like, no matter which 2 pairs, it's always the same rung of the ladder is always the same length. So the whole thing is magnificently homogeneous, or same diameter.
Yes, exactly. Exactly. And but then if you notice that when these bases are interacting, there are these dots that we're seeing, these dashed lines that we're seeing. And this is connoting essentially hydrogen bonding interactions, where, for example, in the top interaction between guanine and cytosine, the very first hydrogen bond is between a carbonyl group extending from the six-membered ring of guanine that's interacting with a hydrogen bound to that's part of an amino group extending from cytosine. There is another hydrogen bonding interaction where a hydrogen from the six-membered ring of guanine is interacting with a nitrogen that is in the heterocycle of that's part of the heterocycle for cytosine. And then we also, with the third interaction, we see again an amino group on guanine interacting with a carbonyl group on the cytosine molecule. And so these are three hydrogen bond interactions. And then for adenine and thymine, we see essentially two hydrogen bonding interactions. And these interactions actually help to stabilize the helix, but more importantly, they're called positional forces, which is a way in which you can ensure that the DNA double helix, again, has integrity in its information, because again, that relationship, adenine always pairs with thymine, guanine always pairs with cytosine. And so those are called complementary base pairing interactions, so that if we know the nucleotide sequence in one strand, we know the nucleotide sequence in another strand. But again, these positional forces actually make sure that the two strands of DNA align perfectly through these, again, hydrogen bonding interactions. And in a hydrogen bond, there is a donor and an acceptor, where the functional group that has the hydrogen is the donor, and then the functional group that has an unpaired electron pair, or unshared electron pair, sorry, an unshared electron pair is the recipient. And so in guanine and cytosine and adenine and thymine, you can see basically the donor and the acceptor pairs there. Now, this is the key to understanding essentially the parity structure that we see within DNA, the parity bit structure that we see within DNA. A few years ago, a chemist by the name of Donald McDonnell at Trinity University in Dublin, Ireland, published a series of papers where he was asking the why question. And his point was, biochemists do a great job of describing the structure of molecules like DNA. So they do a great job of what and how, but very seldom do they actually ask the why question. And so his question was, why these four nucleobases in DNA? Because there are, I don't know, 16 or 20 possible nucleobases that could have reasonably been incorporated into the DNA molecule. Why these particular four? And he argued that, when biochemists do actually address these questions, many times they think about these questions in chemical terms. But his point was, given that DNA is an information storage molecule, could information theory be of any use here? And so he,
But I just want to stop you here, because typically naturalists do not like to ask the why question, because so it seems that this guy is really courageous guy to go against the mainstream. I'm not sure if he's a philosophical naturalist or not. And the second thing is that asking why, and the reason why philosophical naturalists don't like it, because asking why implies that there is purpose, there is a grand plan. And then he even went an extra mile to assume that DNA is an information storage molecule from our perspective, okay? Because in the naturalistic view, it's just a dumb molecule that is guided by completely blind nature through a completely mathematical model of selection. So there is no extremist or radical or fundamental philosophical naturalists would say there is no why, there is no purpose, there is no function for anything. So this guy, now the question is, my question is here, if creation is the true model, then asking why will always lead you to a discovery or to a reason. If naturalism was the real backdrop, then asking why will probably not lead you anywhere, because there is no why. So I'm not sure if you agree with me. This just philosophically hit me that somebody who is studying biochemistry, which is essentially at the end physics, is asking a why question. And
Yeah, you know that's that's really very perceptive, I think, on your part. You know, and you know, to be fair, when you're dealing with biochemical systems, sometimes it's all you can do because of the complexity of those systems is to just simply understand what's going on and how it's happening. And so asking the why question is sometimes a bit of a luxurious type of question to be able to ask. But to your point, that is really, in my mind, the really the more interesting question is, why is it this way? And you're right. For somebody who holds to the view that life stems from the work of a creator, asking the why question is an obvious and natural question to ask. Because that implies, again, there's a rationale, there's a purpose. From an evolutionary perspective, particularly if you're thinking in strict naturalistic, materialistic terms, where evolution is an unguided process, in that situation, asking the why question is almost superfluous. This is just what evolution stumbled upon. This is just what evolutionary happenstance. So you're right. It is on two levels. It's a very courageous question to ask, at least from within an evolutionary framework. I think for philosophical reasons. But again, his curiosity is legitimate. Because there could be other options. A, G, C, and T are not the only options available to us. So why these four nucleobases? And again, to even acknowledge that DNA is indeed an information system, and that information theory may actually give us insight that strict chemistry and physics wouldn't, again, is a very provocative maneuver on this researcher's part.
Absolutely. Let's see if it was rewarding to him.
Oh, it was extremely rewarding, because so what he did was he did, I think, an obvious connection between the hydrogen bonding interactions between the base pairs, and whether it's that bit of information would be a 1 or a 0. It's a 1 if it's a hydrogen bond donor, and it's a 0 if it's a hydrogen bond acceptor. So this is, I think, showing guanine and cytosine. And so you can see how the bits are assigned. And then, just like the parity bit is essentially arbitrary based on the agreed upon protocol, he argued that you could just assign arbitrarily a parity bit to the ring structure. If it is a pyrimidine, which means it's a 6-membered ring, that would be, in this protocol, a 1. And then if it's a fused 5-membered, 6-membered ring, you could call that parity bit 0. And so then you can see how the data or the code word is represented for cytosine, and how it's represented for guanine using that particular configuration.
Okay.
And so this is another diagram, or another image that basically is showing again the hydrogen bonding interactions for not only adenine and thymine, but also for guanine and cytosine. And what he ended up doing in his analysis, if you notice, adenine and thymine only have 2 hydrogen bonding interactions, whereas guanine and cytosine have 3. And so what he did is he said, let's just assume that adenine is actually an amino adenine. So it's a derivative of the adenine molecule. And there you could then get an additional additional data bit. by adding an amino group so that it would be a hydrogen bond donor, where the carbonyl that it doesn't have a pair would become the acceptor. So that would make that position a 0, and then you'd have an additional 1 in the adenine. So that was just a maneuver that he did, where he referred to that as an idealized adenine as opposed to just a real life adenine. And then he did the same type of assignments for other potential nucleobases that could have been incorporated in DNA. And
other than the standard 4.
Yes, right. So he tried different combinations, and what he discovered is that this particular set, adenine, thymine, guanine, and cytosine, are unique in that they form an even parity bit relationship, right?
Wow.
Where we have this even bit parity relationship. Everything else would be mixed parity. These would be mixed parity, and then there are some that actually form odd parity, but what he pointed out is that those particular nucleobases actually are less chemically stable than adenine, thymine, guanine, and cytosine. So these are not only chemically stable, but they form, again, an even parity. These are even parity bit words. And and then or code words. And then he noted that not only do you have hydrogen bonding interactions that take place that attract adenine and thymine and guanine and cytosine, but there's actually, if you have a mismatch, what ends up happening is those hydrogen bonding interactions become not attractive, but repulsive, and therefore force a distortion in the double helix. For mixed parity bit systems, you don't get that repulsion that takes place. If the bases are mixed parity words or code words, then you don't get that repulsion. And so he discovered that representing, again, these nucleobases as, strings of data where you have a parity bit relationship, that again, it's only either the even parity bit words or the odd parity bit words that actually create that repulsion. And it turns out that those nucleobases that will give you odd parity bits are, again, chemically unstable. So in other words, there's this deep-seated rationale for why these 4 nucleobases are found in DNA. It's not an accident, but there's an exquisite, not only chemical logic, but actually a logic that is embedded in information theory. And so to me, this is mind-blowing that there's this level of sophistication in terms of the structural makeup of DNA, where even these hydrogen bonding interactions are not just happenstance, but there's this elegant rationale that is undergirding, these interactions. And so anyway, this is, to me, again, mind-boggling, and it's the type of thing that the more you think about it, the more amazing it becomes. At least for me, it's the type of thing that as I'm laying in bed at night, trying to fall asleep, and I start thinking about this, and my mind begins to wander. It's just mind-blowing to me that this is, again, found in the structure of DNA, is this relationship, but it would be unknown if, again, it wasn't for thinking about DNA in an informatics standpoint as opposed to as simply strictly a chemical system.
So this is truly astonishing. So the question here is, when there is a parity mismatch, the receiver would typically reject the signal and ask for a resend. What would happen? What would happen in the case of DNA if, for example, the wrong nucleotide is incorporated? So instead of a C with a G, 2 C's come together, or I don't know, a C and a T or something. What is the response of the system to that mismatch?
Yeah, well, in that case, you would have essentially it would cause a distortion of the double helix, because when there's a mismatch, that means you're going to get these repulsive interactions as opposed to attractive. In other words, instead you're going to have two hydrogen bond donors interacting, or two hydrogen bond acceptors interacting, and that creates repulsion. For the hydrogen bond donors, it would be something called a steric repulsion, where that's just a word that chemists use, meaning two atoms can't occupy the same location in space, right?
Yeah.
And so when that happens, it things repel. With the donors, these are unshared electron pairs that bear a localized negative charge, and that's going to create a repulsion. But it's because that repulsion is created because that you have either all of these are even or odd parity bits. If they're mixed parity bit relationships, then that repulsion isn't going to happen, because even if you get a mismatch, you're still going to have hydrogen bonding acceptor and donor relationships that you wouldn't have, that even though they're not what's intended, they are still sufficient enough that the helix will remain more or less intact. But that repulsion will cause the helix to actually become distorted, and as a result of that, now it's something that the cell's machinery can detect as an error. Oh, there's a distortion in the double helix. This signals repair enzymes to come in and replace the mismatch base.
Is that also one of the reasons that while replication is happening, if a misincorporation happens, the error detection mechanism can immediately stop and kick the nucleotide out and bring the correct one in?
What's interesting is the enzyme that does the replication, it's called a DNA polymerase, has what's called proofreading function.
Proofreading polymerases have several checkpoints to prevent incorrect nucleotide incorporation during the DNA extension process. The first checkpoint is a result of the enzyme's significant binding preference for the correct versus incorrect nucleoside triphosphate during polymerization. If an incorrect base does bind to the active site, incorporation is slowed, increasing the opportunity for the incorrect nucleotide to dissociate and a correct nucleotide to bind. Alternatively, if an incorrect nucleotide is incorporated, the proofreading polymerase detects the perturbation caused by the mispaired bases and shifts the 3' end of the growing strand to the polymerase's proofreading exonuclease active site domain, where the 3' to 5' exonuclease activity removes the mispaired base. With the mispaired base removed, the polymerase shifts the strand back into the polymerase domain and continues adding bases and extending the DNA.
It improves the fidelity of the replication. But then once the newly formed helix begins to form, if there is a mismatch, it'll create a bulge or a distortion in the helix that can then, after the fact, be detected and then the error correction can take place. So
what you're saying is that even if it escapes the proofreading because of this repulsion
yes,
due to the mixed parity, then another mechanism will still detect it and fix it anyway.
Yes, that's right.
Wow.
That's right. So anyway, it's remarkable. And, to me, as we alluded to at the top of the program, the way I like to think about the design argument is it following the footsteps of William Paley and the watchmaker argument. And I know that argument is oftentimes, maligned, but I actually think there's a genius to it, because Paley's pointing out that, we have from our common experience, we readily recognize things as being designed. And the reason why we recognize those as being designed is because those things that we design have certain attributes, right? And so likewise, when it comes to, again, biochemical systems, I think we can intuitively recognize attributes of those systems that bear similarity to the type of attributes of the systems we design. So I really like the watchmaker argument. It's a simple but yet an elegant but very powerful argument. Well, as you were mentioning when you talked about, the data being sent from the sender and a recipient, that there's things that you have to agree upon, right?
Yeah.
before you even can communicate. So it's not just simply that there is, a string of information like Shannon information. It's actually semantic in that you've agreed ahead of time what constitutes information and what doesn't, and how to detect that, right? But in other words, there are minds that are behind information. Whenever we encounter information that's bona fide information, we realize that information comes from a mind, right? That if it's semantic information, not only does it mean that there's, again, a mind that's responsible for that information, but again, there has to be some element of ascribing meaning, to that information, which again involves the work of a mind. And so the fact that we see DNA as an information molecule in and of itself, to me, is sufficient to say this is probably the work of a mind. But now let's go one step further. And when you realize that the structure of DNA, or that the structure of that information in DNA, it bears similarity to the way that we would structure and manipulate information, takes that argument one step further. It's not just simply that there's information, but that information is organized remarkably in the same way that we would organize information. And information theorists that have studied problems in molecular biology have noted that there is this eerie similarity between the way human language is structured and the way that information is structured in DNA. But now we're going one step further and saying, but even when we think about, how a computer scientist, how an information theorist, how a communication specialist would manipulate and handle that information, to see that similarity is, astounding, and the sophistication of having, a parity bit structure, to the information, to me is amazing.
Yeah, it just reminds me while you're talking, in Christianity, and actually also in Christianity, in I think it's in the Old Testament, there is like God created humans in His image.
Yeah.
And in Islam, it's like خلق الله آدم على صورته, which is almost the same. God created Adam in His image. And I believe that this is metaphorical, and I trust many Christians also think the same, that it means that their attributes of God are the ultimate perfection of certain attributes, and humans are, a miniature of that. So God is the most wise, but we have some wisdom still.
Yeah.
God is the best planner, but we do planning still. And for some reason, God has created us in a way that we follow His ways in a human way.
Yep.
So we get out the best of our minds into how to structure information. And then when we look into His creation, we find the ultimate wisdom of how to structure information. So I think this is telling of something, particularly that His messages essentially communicated that to us, that look into my creation and you will know that I'm there one way or the other.
Yes. And that's exactly how I would think about it, and, I'll use language that a Christian would use, but I think your point is well made, that it's a concept that I think is generalized in both Christianity and Islam, and that is that God is making us in His image. We are in a sense mini creators. God is the ultimate creator, and we're mini creators. And because we bear His image, we can't help but to create things in a way that echo, what that creator has done. It's imperfect, but to me, again, it's astounding that, if you think about, we talked about this in the last episode, parity bits are a recent invention. Is it Hamming that was the kind of the father of error detection, or I don't know.
Yeah,
I'm a biochemist. I shouldn't be.
Yeah. Hamming codes can also actually make self-correcting information transmissions. But even in DNA systems, we see that they find the errors and they correct them actually. But
yeah, right.
Sophisticated ways.
That may be another episode, is to look at the relationship between Hamming
Yeah.
codes and what happens in DNA. But anyway, the idea of a parity bit is, to me, as a biochemist looking on from the outside into the world of information theory, it's a very clever, a very simple and elegant but clever way of detecting error, right? And it's a relative recent invention, but yet here we see that this is a system that was already in place, at the dawn of life's emergence. And it's interesting because in one of the articles that Donald McDonald wrote, he comments that, well, maybe evolution could have stumbled upon this parity bit relationship because it's a fairly simple way of doing error detection.
Is it? Come on.
But here's another way to think about it. Even if you are viewing life's origin in evolutionary terms, it's like you won the lottery, right? It's like, okay, so we just happen to have, of all the possible nucleobases, the 4 that are selected just happen to be the ones that are the only 4 that give us this, even bit parity, that form this even bit parity code words. And and they also, by the way, happen to be the most UV stable bases, and they're also the most chemically stable bases, and they're the only 4 that will allow us to detect error if it happens. It's like everything just, you got very lucky by all these coincidences just stacking up.
I actually, as an engineer from the other side of the table, looking at those kinds of arguments, maybe evolution stumbled on over this or that, I really don't buy it. I'll tell you why. Because as we were saying in the beginning, when we have a sender and a receiver, if the sender decides on his own that he is going to use a new mechanism that he is going to call parity check, okay, and then he reserves a bit to do the parity check. So now there is 7 bits of information, and then the 8th bit is parity. And then he concludes that there must be 2 systems of parity check, one even and one odd parity, and he picks one of them. How on earth would the receiver know any of this? So the receiver can go on considering that all 8 bits are essentially information and there is no parity. And if he, one way or the other, decides that there is parity, he gets the revelation in the same day or moment, he wouldn't be able to guess what kind of parity checking is being used on the other side. So it just doesn't make sense, because when there is an integrated system, I like to call this integrated systems. Maybe, like you said, some like Dr. B, he would like to refer to that as irreducible complexity or what have you. But at the end of the day, there is this kind of conspiracy between all parts of the system that if it happens by any whim of chance on one side, it is not conceivable that on the other side, the other guy just has the same ideas and the same protocol is established by chance. This is absolutely not possible unless somebody would go to the absurd proposition that all of those intricate systems go into this unbelievable, it's winning a one in a billion lottery a billion times in a row. It just keeps winning the lottery every single time, not on one side, but all the conspirators are winning the lottery in the same piece of lottery. It's completely absurd.
Yes, but that in effect is what the Donald McDonald was saying in his article, though I don't think he was recognizing the absurdity of it. But it's just that you're getting lucky time and time again, the coincidences keep stacking up, And at what point do you say something has been rigged here? There's
Yeah, enough
something
is enough.
Yeah.
Absolutely. Fuz, that has been really amazing. And we'll have some references for the audience in the descriptions about papers by the scientists you mentioned, and if anyone wants to do some further reading about the topic. But we are also going to have the after show right after this, so you'll find the link to it in the description and also in the live chat. So jump over there if you have further questions about this topic. And thank you very much, Fuz, for this. I hope everybody has seen how this is really glorifying God's creation and showing us how to see God in biology. So God bless you all. And thanks a lot, Fuz, and for this. And see you on the next episode. And prior to that, in the after show, be sure to send us your questions also for the next episode. And see you again, Fuz, soon, next Thursday.
That's right.
All right. So for everybody, have a great time on the after show, and let's do this again next Thursday. Assalamu alaikum. Peace be upon you all.